In this video, we're going to be looking at nucleic acids, comparing the similarities and the differences to the different types of nucleic acids there are. Okay, there are two kinds of nucleic acids. The first one being DNA, which is deoxyribonucleic acid, and the second one being RNA, which is ribonucleic acid. Now, both of these are polynucleotides. Now, polynucleotides just means that we have lots of nucleotides that have been joined together and it forms a long chain. Now, a nucleotide is a sugar, which is deoxyribose in DNA, or a ribose sugar in RNA. Okay, it's made up of a sugar, a nitrogenous base, so A's, T's, C's, G's, and a phosphate group. So there are four different bases in DNA: adenine, which is A, thymine T, cytosine C, and guanine G. Now, one really important thing to notice is that with RNA, the thymine, the T, is replaced by a base called uracil, which is a U. Now, uracil always base pairs with adenine, as thymine always would in DNA. So it pretty much just means that uracil is replacing thymine and it still binds with adenine. Okay, so in DNA, two polynucleotide strands join together by hydrogen bonds. Okay, basically we have these bonds between the two bases, and it forms that bridge between the two strands, the leading and the lagging strand, and then that one actually makes a double helix in the DNA. So each base can only join with one specific base, A's with T's and C's with G. We call this complementary base pairing. Okay, so we've got our nitrogenous bases here. Okay, this one here would be A and T, and the reason why we can see that is because A and T has two hydrogen bonds between the bases, as we can see here, 1 and 2. Cytosine and guanine have 3 hydrogen bonds, as we can see, 1, 2, and 3. So this one here would be cytosine and guanine, the same here. Okay, and this will be adenine and thymine because of these bonds. We've got our sugar phosphate backbone here, which is our sugar, deoxyribose sugar, and our phosphate group, and then our nitrogenous base that makes up the backbone of DNA. Okay, so we know that DNA codes for everything in life. Okay, it codes for certain characteristics. Now, DNA makes up genes. Now, a gene is a section of DNA that codes for proteins. Okay, it's found on chromosomes. Now, genes are instructions for proteins. It's basically meaning that DNA itself is the code and it's creating proteins. Proteins are made up of amino acids. Okay, so basically amino acids are produced from the DNA, which is the code, and lots of these amino acids join together to form proteins. Now, the order of amino acids determines what type of protein it would be. Okay, in a gene, it is the order of bases in the DNA that determines the amino acid, which amino acid is made. So each amino acid is coded for by a sequence of 3 bases, and we call this a triplet or codon. Now, it basically means that amino acids are produced from that template strand. 3 bases we call a triplet or a codon. Okay, we'll look at that very shortly. Different triplet codes for different amino acids. And so the sequence of triplets in a section of DNA acts as a template for the production of a protein. Okay, so RNA, what is RNA? So RNA is quite similar to DNA. It contains the uracil instead of the thymine, as we just talked about before. One really important thing to understand is that DNA is a really large molecule, and it is quite difficult for the molecule itself to get through the nuclear membrane. Now, the reason why this is important is because in order for us to make proteins, we have to have a copy of the DNA molecule itself, and then that molecule can actually be transported out of the nucleus through the nuclear membrane so that it can be synthesized, or proteins can be synthesized from that code. Okay, so DNA cannot get through the nuclear membrane, so smaller RNA is used to copy a section of the DNA, and this is called transcription. So just imagine DNA as being an ancient book. Okay, we want to create a copy of that ancient book so we can create proteins from it. RNA is our photocopier, basically. It's how we're making the copy. So RNA is making the copy for transcription. The RNA then leaves the nucleus and joins with a ribosome, where the protein is then synthesized. Now, the organelle ribosome is for protein synthesis, and we call this translation. So transcription and translation, the two steps for protein synthesis. Okay, so apart from uracil, there are two differences with RNA compared to DNA. The first thing is that the sugar is a ribose and not a deoxyribose. Okay, the second thing is that the nucleotide strand is single-stranded and not double-stranded like in a DNA molecule. There are a few different types of RNA, one of them being mRNA or messenger RNA, and the other one being transfer RNA or tRNA. Okay, what we'll do is we'll look at two of these different types of RNA now. Okay, so this is what RNA looks like. Okay, we can see it quite clearly that the uracil takes place of the thymine. So we've got a single-stranded RNA molecule with the uracil here. Okay, A, G, U, and C. We don't have anything complementary base paired to these bases because it is single-stranded as RNA. Okay, so mRNA is single-stranded, okay, because it's RNA. Groups of 3 bases are called codons. Now that's really important when we're looking at mRNA, because mRNA is actually that transcript of the DNA molecule itself, or that copy, and 3 bases of the messenger RNA is the codon, which is actually coding for the amino acid. So mRNA is made in the nucleus during transcription and takes the genetic code out into the cytoplasm, where the ribosome is used to make a protein during translation. Okay, we can see here this is the first step with transcription, and this is occurring inside the nucleus. Okay, we've got our DNA molecule here, and we've got messenger RNA, which is complementary base paired to the DNA bases. We can see here G, C, A, U. Okay, no thymine, we've got uracil. Same here, A, U, CG, AU, and then thymine for the DNA, and adenine for the mRNA. Okay, we'll look at tRNA or transfer RNA. Now, it is completely different, this RNA molecule. It is a single polynucleotide chain, which means it is single-stranded, but it has a different shape. It's in a clover-shaped, it's folded into a clover shape, and this actually allows for certain base pairing between the messenger RNA codon group, okay? And we'll talk about that. So it is held in the shape by hydrogen bonds between the bases AU or CG. At one end of the transfer RNA molecule, there are specific sequences of 3 bases called an anticodon. Now, the anticodon is actually what is binding to the codon itself from the mRNA. Anticodon just means the opposite for the codon. So at the other end is an amino acid binding site. The tRNA carries amino acids to the ribosome where they are used to make proteins. Okay, so this is the tRNA molecule itself. We can see that we have got a clover shape molecule with the bonds between the C's and G's and A's and U's in between here, which is giving it that specific shape. We can see we have the codon, which is from the mRNA strand down the bottom here, okay, and the anticodon is what is complementary base pairing to the codon. So the anticodon is found on the tRNA and the codon is found on the mRNA. Now these 3 codons, the triplet code here, codes for a specific amino acid. Okay, and the amino acid will actually be formed up the top here, which is attached to this group here. Okay, and that's it. So there are 3 different types of nucleic acids, one of them being deoxyribonucleic acid or DNA, and the other one being messenger RNA and transfer RNA.